Hello everyone, this is Dr. G. Anita Hebsiba, Department of Chemistry, Dr. S.N.S. Rajalakshmi College of Arts and Science, Coimbatore. In this video, we are going to discuss about enzymatic desizing. We are very much familiar with the word desizing. It means the removal of size from the textile material that is cotton. This desizing can be achieved by many ways, by rod steeping, by acid desizing, by caustic soda desizing, oxidative desizing, enzymatic desizing, plasma desizing, etc. Uh, this video will focus on enzymatic desizing. Normally, when we use hydrochloric acid or caustic soda or water as a method for desizing, it may result in damaged fabric because these chemicals and also the action of water for a prolonged period on the cotton fabric may damage the fabric. So in that perspective, uh, those disadvantages which are highlighted for rod steeping, acid desizing and caustic soda desizing is the need for enzymatic desizing. What is this enzymatic desizing? It is the hydrolysis of starch using enzymes under particular concentration, temperature and duration. Enzymatic desizing is the most widely experienced method to desize the starch nowadays. Earlier we were practicing acid desizing, caustic soda desizing, oxidative desizing and all, but nowadays enzymatic desizing is common in practice. In enzymatic desizing, enzymes liquefy the starch in the fabric. Actually, starch is coated on the fabric. It is insoluble. We have to make the starch soluble and we have to remove it during the wash process. This enzyme which is being used for desizing will liquefy the starch. That means the insoluble starch is being converted into soluble starch and it can be easily removed. But what is an enzyme? An enzyme is essentially a protein produced by living cells. They are all biocatalysts which are used in chemical reaction. By the word catalyst, we mean that they are added in very small quantity, they speed up the reaction and they are regenerated at the end. So here also enzymes are used in very small quantity and they speed up the reaction. So, and these enzymes are very specific in reaction too. They are very sensitive. Uh, they require the very correct temperature to be maintained, pH to be maintained, and all the conditions to be maintained properly. Here, when we come to enzymatic desizing, usually we take malt extract as the enzyme, that is 0.5 to 2 percentage. Chelating agent or salt if necessary. What is a chelating agent? This chelating agent, they are all sequestering agents or else complexing agents which will remove the metal ion which is interfering in the enzymatic hydrolysis process. So for example, if the water which is used for the enzymatic hydrolysis process contains calcium ion, it may interfere with the activity of the enzyme. But the chelating agent will form a complex with that particular metal ion and hence the action of metal is deactivated. So that is the role of the chelating agent if it is necessary and if the water bath contains or else the desizing bath contains calcium ion, chelating agent or salt is added. And what about the temperature? It is about 50 to 60 degrees Celsius. It has to be maintained. Time is about 3 to 4 hours and the pH is 6 to 7.5 hours. We were able to reduce the time from 12 to 24 hours in the case of rod steeping to 3 to 4 hours and also eco-friendly method. How this desizing is carried out. The sized fabric is in the form of a rope, that is rope form, and it is immersed into the desizing liquor by immersion roller. The consumption rate should be regulated so that the mixer level remains constant, or else initially when you dip, the fabric will absorb more desized liquor, and later when the fabric comes in, we won't be able to maintain the level. So the consumption rate should be regulated, and then the fabric is squeezed and stored for 3 to 4 hours at room temperature. Here enzymes react with starch and decompose into glucose. What are the advantages and disadvantages? Here we do not use any aggressive chemicals, so there is no damage to the fiber. Wide variety of application processes are available with the enzymes. What are the disadvantages? Lower additional cleaning effect towards other impurities. You cannot expect 100% scouring or else 100% desizing to be done using enzymes. And also tapioca starch cannot be removed by enzymatic desizing. Thank you.